بسم الله الرحمن الرحيم. أعزائي طلبة المرحلة الثالثة كلية الطب جامعة النهرين. اليوم إن شاء الله راح نتكلم حول موضوع الكانسر immunology, tumor immunology and immunotherapy. راح أحاول إن شاء الله أشرح لكم إياه بشكل فيديوي على الباوربوينت اللي أرسلت لكم إياه سابقا، عسى أن تتوضح بعض الأمور إن شاء الله. وأي سؤال إن شاء الله ممكن تراسلوني على الجوجل كلاس روم ونتواصل إن شاء الله. Tumor immunology. First, we should know some basic knowledge of cancer. What is cancer? How does cancer occur? How many types of cancers? And current therapeutic strategies for cancer. You know, cancers consist of single clones or several clones of cells that are capable of independent growth in the host. And cancer cells arise from host cells via neoplastic transformation or carcinogenesis. There are many carcinogens, physical, chemical, biological agents that may cause cancer individually or in corporation. Some example of some carcinogens are radiations like ultraviolet light, sunshine, X-rays, radioactive elements. All these can induce DNA damage and chromosome breaks. There are some chemicals like smoke and tar. They can damage the DNA and cause mutagens. And there are some oncogenic viruses that they insert their DNA or the copy of the DNA of these viral oncogenes into the genome of host target cells. And there are some hereditary factors like some oncogenes are inheritable. There are active growth regulatory genes like growth factor receptors, molecules of signal transduction and transcription factors referred to as oncogenes. There are some genes that inhibit growth. They are called suppressor oncogenes. And there are some genes that regulate apoptosis like BCL2, PAX, and BID. I think all these basic knowledge you know from the pathology side, from the pathology semester. The classification of cancer, they usually are classified into carcinoma. Carcinoma means epithelial origin involving the skin, mucous membrane, epithelial cells in the glands. Sarcomas, they are cancers of connective tissues like the liposarcoma, fibrosarcoma, etc. Other type of cancer is the lymphoma, which means the cancer of the tumor of the T cell, B cell lymphoma, Hodgkin's lymphoma, Burkitt's lymphoma. They are usually solid tumors. And there are other type of tumor, which is the leukemia. It is usually disseminated tumor because it is cancer or tumor of the blood cells, either from the lymphoid or myeloid region, acute or chronic, ILR, CML, and etc. There are current strategies to combat the tumors. These strategies involve the mechanics, which involves the surgery. It's very old and still useful. There are other strategies to combat tumors like the radiotherapy, which is physical. There are some chemicals, which is the chemotherapy, started from the 1942, and there are biological strategies to combat a tumor, as in immunotherapy, and it started in 1976. What are the advantages of immunotherapy for cancer, which is the important subject that we are related to immunology? First, you should know tumors are immunogenic. Second, they kill the single cells. They are not killing all the other cells, while in cases of chemotherapy, not the immunotherapy, usually it causes damage to other types of cells, the replicatory cells. Usually they migrate to the tissue, this kind of immunotherapy. There is memory of this immunotherapy usually to that type of cancer, which is very useful if there is recurrent of that cancer. It is usually specific, so there will be no damage to other types of normal cells. As in chemotherapy, usually there is damage to other cells like the mucosal cell of the GIT, for example, the damage to the blood cells and causing decrease in white BC count. Patients become vulnerable to infection more and decrease in RBCs, etc. And the other advantage of immunotherapy of cancer is the lifelong protection, because usually it's related to the memory production of that immunotherapy. Now, to understand the tumor immunology and immunotherapy, we should put these questions that we will try to answer them during these two lectures. First, how does our immune system eliminate cancer cells? Second, how usually this is in our daily life. Second, how do cancer cells escape from immunosurveillance that we will talk about? Third, how can we manipulate the immune response to kill cancer cells? And the current approaches of tumor immunotherapy with some examples. To answer the first questions, how does our immune system, this is the first question, how does our immune system eliminate cancer cells? This is called tumor immunosurveillance, المسح المناعي للأورام. The hypothesis was first proposed by Thomas and Burnt in 1957. Immunosurveillance, it means the immunological resistance of the host against the development of cancer cells. Immune response against tumor cells take place at an early stage in tumor development that will lead to eradicating the cancer before it becomes apparent. This is what is meant by immunosurveillance. It means that our immune system is usually performing an immunosurveillance daily to detect any abnormal tumor cells and eradicating these cells before it become apparent. And this happens daily in our body. Usually about a lot of cells can change into cancer cells, but they are detected early during their change, and our immune system will kill them. And this is very important to our body. This is what is called the tumor immunosurveillance. طبعا the tumor immunosurveillance you should put in mind is usually performed by many ways, and it happens daily in our system. Subhanallah, there are thousands of cells that change into tumor cells daily, but our immune system can eliminate them. Now, what are the weapons of the host? First, there are antibodies. There are natural killer cells, there are dendritic cells, some cytokines, chemokines with the help of T cells and the macrophage, in addition to the cytotoxic T lymphocytes. All these cells will act together in order to kill these tumor cells at the early stage of their development in the immunosurveillance way. Now, starting with the antibody, usually the mechanism by the direct attack. Usually these antibodies will block the growth factor receptors. These are growth factor receptors responsible for the growth of the tumor cells. When they are blocked, will lead to arresting their proliferation of these tumor cells, or will lead to apoptosis, inducing apoptosis, which is the regular killing of these abnormal cells. Usually it's not sufficient to completely protect the body, so there is the indirect attack of these antibodies. This is the second way. It is usually the major protective effort. عندنا هنا two terms. أول term هو ال ADCC والsecond term هو ال CDC. ال ADCC is the antibody dependent cell mediated cytotoxicity. It means the recruiting cells that have cytotoxicity such as monocytes and macrophages. From the name, it is killing of the cancer cells by a cell-mediated cytotoxicity. Usually it is antibody dependent. And the other way, by killing these tumor cells, is called the CDC, which is the complement dependent cytotoxicity. احنا هسه في وقتهم سبحان الله هذا هو السي دي سي، مصطلح يعني يشابه مصطلح آخر اللي هو Central of Disease Control or of communicable diseases، اللي هي مركز السيطرة على الأمراض الانتقالية. وإحنا في وقت هذا السي دي سي قد يكون المصطلح شائع باعتبار موضوع الكوفيد 19. هنا اللي نقصد السي دي سي هو الكومبلمنت ديبندنت سايتوتوكسيسيتي. It means the binding of receptor initiating the complement system, the complement cascade. واحنا حكينا سابقا في قاعة المحاضرات في الكلية، وحضراتكم كنتم حاضرين على الكومبلمنت كاسكيد، وعدنا هذا الموضوع resulting in membrane attack complex اللي هو الماكس اللي متكون من ال C9 والother components of the complement, causing cell lysis and death. This has happened not only to the microorganism but also can happen to the abnormal tumor cells in our bodies. So these two methods of antibodies, the direct attack and the indirect attack, and the indirect attack is by two ways, the ADCC and the CDC. The other weapon is the cytotoxic T cell, which is the CD8 positive T lymphocytes. They are attacking or attaching to class 1 MHC, class 1 major histocompatibility complex, the peptide complex that is present on this MHC. They destroy cancer cells by performing, leading to perforation of the membrane with an enzyme, or by triggering an apoptotic pathway, release of an enzyme. If there is any abnormal peptide that is presented by cells, abnormal cells that carry MHC class 1, will lead to the activation of cytotoxic T lymphocyte and will lead to perforation of that cell or triggering an apoptotic pathway. What is the role of the helper T cell, which is the CD4 positive T lymphocytes? It's usually these cells reacting into class 2 MHC peptide complex, and they will secrete cytokines. These cytokines will lead to, if it form type 1 helper T lymphocyte, T helper 1, it will lead to cytotoxic T cell response. It will activate the CD8 positive T lymphocytes, while if it's the T helper 2, so it will lead to antibody response, will divert the way into an antibody response. And we saw what's the role of the antibody by direct and the indirect way, and the indirect way by two ways also from the previous slides. Now, what is the role of cytokines? Cytokines are very important also in the immunosurveillance. Usually the cytokines، واحنا اخذنا محاضرة الcytokines نعطيناكم اياها في بداية المقرر. Cytokines usually regulating the innate immune system, the natural killer cells, macrophages, neutrophils, and the adaptive immune system, the T and B cell. عندنا example هو الانترفيرون الانترفيرون alpha. Interferon alpha usually will upregulate the MHC class 1 tumor antigens and adhesion molecules. The molecules اللي تسوي adhesion will lead to promoting activity of B and T cell in addition to macrophages and dendritic cells. Another example of an important cytokine is the interleukin 2, which is the T cell growth factor that binds to a specific tripartite receptor on the T cell. The interleukin 12, it will promote a natural killer and T cell activity and the growth factor of B cells. وعندنا الجرانيولوسايت مونوسايت colony stimulating factor تذكروا هذا هم أخذيه بمحاضرة السيتوكاينز reconstitute antigen representing cells. It will help in that pathway. The dendritic cells also are important. They are professional antigen presenting cells in the final common pathway for activating the naive T cells and it is important in immunosurveillance. Now, after all these mechanisms, a question will come to your mind maybe. Then if there is immunosurveillance and the immune system can kill these cancer cells, how does the tumor progress? How does tumor progress? طبعا الموضوع هو إذا الimmune system can detect these single tumor cells بس إذا كان الجروث مالتهم from these single tumor cells will go uncontrolled or undetected by the immune system will lead to the advanced tumor and the tumor can be presented clinically. Now, the fortune of the tumor cells will depend on the battle between immune system and the tumor cells. Which one of them will become victorious? It will lead to the outcome. If the immune system is the victorious, the tumor cell will regress. If the tumor cell will win this battle, it will progress into tumor mass. These things depend on the first weapons from the tumors and or a defect of the immune system. مثل المعركة إذا كانت الويبنز مال التيومر قوية أو إذا كان الديفكت بالإميون سيستم بالدفاعات مال الإميون سيستم هي قليلة بالنتيجة the tumor can progress. طبعا الويبنز اللي يستعملها التيومر أو التيومر cells هي many and these are called how tumor cells will avoid the immunosurveillance. These are very important things. These are the weapons of the of the tumor cells. So first, the tumor cells can alter their characteristics. First, either they will lose or downregulate their MHC class 1. يعني بحيث الاميون سيستم تعرفون انه اذا ماكو MHC class 1 او ماكو MHC class 2 there will be no presentation of the peptides of abnormal peptide of that cell and the immune system will not detect them. Second, downregulation or mutation or loss of tumor antigens. These tumor antigens اللي راح ناخذها اكو certain antigens secreted from the tumors they are foreign to our body. So these antigens will be target of the immune system. So if there is loss or downregulation or mutation of these tumor antigens, they will not be detected by the immune system. Or there is another important thing is the loss of co-stimulation. The co-stimulus that is complementary to finish this process of killing of that abnormal tumor cells اللي أخذنا سابقا مثلا في الرياكشن مثلا MHC1 MHC2 عندنا CD40 molecules and their ligands shown they are called as co-stimulatory. If there is loss of co-stimulatory molecules, so there will be the tumor cell will win. Now, the other mechanism is the suppressing the immune response. The tumor cells ممكن تغير their characteristic, but they can suppress the immune system. تأثر على immune response بحيث أنه تسوي له suppression. مثل ineffective signals to the cytotoxic T lymphocytes. Other example is alteration in cell death receptor signaling. أكو receptor signaling causing death of these cells راح يصير بها alteration. Or there are some immunosuppressive cytokines. It will lead to down regulation of immunosuppression of the immune system and therefore راح يكون هذا immune system خامل في مقابل tumor cells and the tumor cells will progress. Or the third reason is the outpacing of the immune response. معناته أنه immune response ما يلحق على سرعة انقسام الtumor cells. tumor cells can simply proliferate so quickly that the immune response is not fast enough to keep their growth in check. يعني إذا كان الجروث cell 1 cell 2 cell here and there other cells وكلها ممكن immune system يسيطر عليها maybe more than this، بس إذا كانت السرعة thousands of cells ممكن تنقسم بسرعة بحيث أنه الimmune system ما يقدر cannot keep enough to their growth in check، ما يقدر يشيكها بشكل منتظم بحيث يعبر عليه other cells and can progress into a tumor mass. Now طبعا هاي كل واحدة ممكن احنا نحكي عليها، هو مثلا loss أو down regulation of HLA من class 1 MHC1 مثل total loss haplotype loss HLA allelic loss compound phenotype، كل هاي example على loss أو down regulation of HLA class 1 MHC class 1. Second is the down regulation or mutation or loss of the tumor antigens. طبعا tumor antigens اللي هي تختصر TA أو عندنا ما يسمى بالtumor associated antigens. Some antigens are associated with the growth of these cells مثل يعني some antigens هي موجودة أصلا بالبودي بس هي ترتفع in certain tumors مثل ال prostate specific antigen PSA وغيرها اللي هو موجود in certain amount، بس إذا صار in amount كبيرة طبعا بالنتيجة ممكن واحد نعتبره كتيومر associated antigens. واكو tumor antigens هي أصلا ما موجودة in our body only present in cases of tumor. There may be either complete loss or down modulation of these tumor antigens or tumor associated antigens. عندنا some altered self antigens ممكن تتحول إلى tumor antigens. عندنا viral antigen مثل البابيلوما فيروس antigen of cervical carcinoma مثل الإبنة وغيرها. oncofetal antigens مثل الألفا فيتو بروتين and carcinoembryonic antigen من الاسم هي related to the fetal life وعادة ما تكون موجودة بالadult life either but in cases of tumors can be present. And there are some autoantigens which are overexpressed as in cases of HER2/neu اللي هو epidermal growth factor receptor in cases of breast cancer and the use of herceptin in this case is important. يعني some of the tumor cells some of the breast cancers who are related to the upregulation of these growth factor receptors, and so there is a way of biological treatment that is using the herceptin blocking these receptors in order to down regulation of these growth these cells. The other way is the loss of co-stimulation as we say عندنا co-stimulatory molecule recognize مثل البي family. عندنا some CD receptor مثل CD40 ligand molecules اللي ذكرنا عليه اللي ذكرت لكم اياه بالexample سابقا. Other examples of loss of co-stimulation will lead to incomplete response of the immune system to the tumor cells and there is no killing of these tumor cells. The fourth is the alteration in cell death receptor signaling with the defects in Fas and Fas ligand signaling system اللي هو an important pathway for the apoptosis pathway. So there will be resistance to apoptosis or there is overexpression of BCL2 and this will lead to apoptosis resistance also, which is regarded as one of the cell death receptor signaling. Immunosuppressive cytokines, the fifth way is the immunosuppressive cytokines. A number of immunosuppressive cytokines like the interleukin 10 مثلا inhibits antigen presentation and interleukin 2 production. VEGF اللي هو vascular endothelial growth factor that will avoid recognition and inhibit the effector function, prevent the T cell activation and the cytokine production. The sixth way is the induction of immunosuppressive cells عندنا مثل CD4 positive CD25 positive T lymphocytes they constitute about 5 to 10% of CD4 positive T cells. They are immunological. They will lead to immunological tolerance to self-antigen and the inhibition of T cell proliferation. And there are other examples like expression of granulocyte monocyte markers accumulate in the spleen, lymph nodes, and the blood of tumor-bearing mice. Example عليه. And inhibiting antibody production, cytotoxic T lymphocyte generation, T cell function, lymphocyte proliferation, and other examples of the induction of immunosuppressive cells. المهم أنه there will be an immunosuppressive cells that will be upregulated, that will be induced by the tumor cells. These are immunosuppressive cells. من اسمها suppressive, they will suppress the immune response, the immune system. 7 is the production of other suppressive factors. عندنا مثل endolimine expressed in most human tumor cells and splenic dendritic cell subsets, leading to blockage of proliferation of T cells. Ganglioside اللي هو sialic acid containing sphingolipids and the prostaglandins. All these will lead to suppression of the immune system, as suppressive factors can be produced by the tumor cells and regarded as a weapon. In summary, the main defenses of the tumor against immunity: first, alteration of the MHC class 1 and the tumor antigen expression. Second, dysregulated expression of adhesion or co-stimulatory molecules by tumor and or antigen presenting cells. Third, change in T cell signal transduction molecule, that's to say cell death receptor signaling مثل ما قلنا بالFAST والFAST ligand pathway. Fourth is the induction of the immune suppressive cytokines. Fifth, induction of the immune suppressive cells. And the sixth is the secretion of suppressive factors. Some suppressive factors, all these will lead to the proliferation of the tumor cells and the immune system will not be able to destroy these tumor cells.